Halsey. This video is an introduction video to our blood typing lab. So first of all, I want to talk about what do the blood types mean. We're going to be talking about A, B, and O blood typing. There are other blood types, but we're not going to discuss those. And these are the ones that people are commonly concerned with when it comes to donating blood and getting blood in a blood transfusion. So you can have blood type A, blood type B, blood type AB, or blood type O. Those people that have blood type A have a particular protein on the surface of their red blood cells, and the people with type B blood have a different protein on the surface of their red blood cells. People with type AB blood have both types A and B proteins on the surface of their red blood cells, and people with blood type O don't have either. Now, if you have blood type O, you produce antibodies for blood type A and B, so that if you get a transfusion from somebody with type A, B, or AB blood, your body will attack it. If you have blood type B, you make antibodies for A, and that means that your blood will attack the blood that is transfused into you. If you have type A, you make antibodies for the B protein. So the idea is that if you are type AB, you don't make any antibodies because neither of those proteins are foreign to you, because you were born with both of those proteins on the surface of your red blood cells, so there's no foreign proteins. The idea is that if you get a blood transfusion from somebody with incompatible blood type that has a protein on the surface of their blood that is not a protein that you were born with, your body will recognize it as a foreign invader and attack it, and that can actually kill you. Some of the symptoms of ABO incompatibility reaction, which is a reaction when you get a blood transfusion with an incompatible blood type, is fever, breathing difficulty, muscle aches, chest pain, and blood in your urine, and often it can be fatal. The interesting thing is when I was looking up the symptoms, the number one symptom is a strong feeling that something bad's about to happen, which made me laugh because it is about to happen. You might die. So in any case, blood type O is the universal donor, which means that anybody can get O blood because it doesn't have any foreign proteins on the surface. And blood type AB is the universal recipient. So someone with blood type AB can get any blood type without having a problem. Now, this is not including the Rh factor. The Rh factor is a rhesus factor because it was discovered in rhesus monkeys, and you're either positive or negative. Rh negative people cannot get Rh positive blood. So in actuality, O negative is the universal donor, and AB positive is the universal recipient. But for our purposes, we're going to focus on AB and O blood types and not really focus much on the Rh factor. I happen to have O negative blood, and so therefore I am the universal donor. They always use O negative blood in emergency situations because they don't have time to figure out what the person who needs the blood's blood type is. But typically they will attempt to use type-specific blood so that they can save the O negative blood. Only approximately 6% of the population has O negative blood. So the blood banks really do need to save the O negative blood for emergencies. And sometimes if you go in for an elective surgery, they will have you bank your own blood to give it back to you in advance. So we're going to be doing a lab with looking at results of a blood typing method called the Eldon Card. So basically you get a little card, and it is coated with like a waterproof coating, and on the Eldon Card they put an antibody to A, an antibody to B, and an antibody to the Rh factor. So anti-D is the Rh factor, and then a control. And what they look for when they put drops of blood on it is a clumping. And that clumping is called agglutination. And the I that you have been using for all your blood typing problems, like the capital I, A, and capital IB, and the two little I's for O blood type, that stands for isoagglutinogen, and it's kind of related to the agglutinate word. So here I'm going to click on a little video showing you somebody actually doing blood typing using an Eldon card. So they're putting a solution on each one. They're now going to put a droplet of blood on in each of these circles from the same individual. So they're testing at the same time to see if you have protein A, protein B, or antigen A, antigen B, and the Rh factor. So you can see it's starting to clump in the B circle, or agglutinate. And if you notice, there appears to be no clumping in the anti-D circle, so that means they are Rh negative. And it looks like it's clumping in both A and B, so they would be AB negative. And they're setting a timer because you're supposed to wait, but you can kind of already see the results. So it looks totally normal in the control, and the Rh factor, no, yep. Well, I can't tell. It sort of looks like it's clumping in the Rh factor one. Maybe they are AB positive. Yep, it looks like they're AB positive. So if you look at these Eldon cards, this one had a reaction to protein A and a reaction to the Rh factor. So this one would be A positive. So they have type A blood and they're positive for the Rh factor. And this one would be A negative because it clumped in A, but nothing in the Rh factor. If it clumped in none of them, it would be O negative. If it clumped in all of them, it'd be AB positive. So this is the file of the lab that you're going to be doing, and just for the record, the little squiggles are showing you the clumping. So this one is clumping with antibody A but not antibody B. This one is clumping with antibody B and not A, and this one is clumping with both. So you should be able to figure out what the blood types are. Be really careful here for phenotypes. You're going to say the name of the blood type, A, B, or O, and here under genes and possible genotypes, you're going to use the notation with the I's. Please don't switch these. So in this lab, there is a scenario where there are 3 babies and 3 couples, and there's a mix-up at the hospital, and you have to figure out, based on blood types, whose baby is whose. Certainly now we could use DNA fingerprinting, but imagine if this had happened before that technology. And also, blood typing is actually very cheap to do. So while you certainly can do DNA fingerprinting, you can rule out possibilities using blood type and save a lot of time and money, because blood typing is very fast and very inexpensive. So basically what you're going to do is you're going to, using the parent's blood type, figure out all the possible blood types that the babies could be, and then you're going to figure out which baby is which. So you need to make sure you're going to show your blood type Punnett squares by inserting a data table. There's like a little thing that looks like, well, it's a little, this is Word, but you're going to be doing it in a Google Doc. But there's a little icon that looks like a data table, and you're going to do a 2x2 data table, and then you type in it. So then it asks here, how do you figure out whose baby is whose? Students always ask me, can I say process of elimination? Only when you get to the third one. You can't start from the beginning. But by the time you get to the third couple and the baby, then you can say process of elimination. So let's kind of discuss the analysis questions. So question number 1 says, if a baby is left on a doorstep of a church and the mother changes her mind and comes back and wants to reclaim her baby, can the mother come back and say the baby is hers based on blood type alone? So this is not in a scenario where they can eliminate it to a few couples and use process of elimination. This is like, let's say I had a baby and it has blood type O negative. I left it on a doorstep. Can I come back and say, hey, that's my baby, and I can prove it because I'm O negative and so is the baby? People usually have an easy time with question number 2. Question number 3, I get a lot of questions about. It says, what can be proven in issues of paternity and maternity using only blood types of parents and babies? So can you prove somebody is the mother or father? So paternity means father, maternity means who's the mother, using blood types alone. Can you prove or disprove who the parents are and explain your answer? And this means without DNA testing. Question number 4 is fairly straightforward. For this last question, this actually came from a newspaper article. There was an advice columnist who was called Ann Landers, and her sister was also an advice columnist called Dear Abby. Dear Abby was maybe a little bit more famous. Neither of them are still around. This was, they've been around since like the 60s, I think, maybe the 50s. But this was in the newspaper in 2002, and I cut it out because we were right in the middle of the genetics unit. But it's a letter to Ann Landers about a baby that was born and not having the same blood type as her or her husband. So you're supposed to read this article or this little clip and then tell me what is wrong with Ann Landers' response to her, and you write a short explanation and you fill in the Punnett square. You get a certain number of points for the explanation and a certain number of points for the Punnett square. There are situations like you can actually do 2 possible Punnett squares, but if you do the right one, you only need 1. Also, students also get hung up on the fact that it says be positive. Ignore the positive and negative for the RH factor in this scenario and only focus on the A, B, and O blood types, and that's enough information to really answer this question. So you don't need to address the positive and negative for RH factor in the blood type.